We've now reached chapter 44, which is all about osmoregulation and excretion. What you'll find within this chapter, as is often the case within the Campbell book, is that there is plenty of discussion about how osmoregulation and release of waste occurs within different types of organisms. We'll go through all the different organisms that your book goes through. Where you want to place a ton of emphasis, however, is going to be on mammalian osmoregulation, the mammalian urinary system. That one is a little bit more detailed within your book, and you do want to pay attention to the difference in terms of weight between these different topics. Concept 44.1. Osmoregulation balances the uptake and loss of water and solutes. So when we think about osmoregulation, we'll definitely be incorporating our discussions of topics that were relevant within Chapter 7. And that should make sense for what we're talking about right now, because we're really thinking about movement. And that movement incorporates both water and solutes. So the process of osmoregulation is going to incorporate proteins and aquaporins and discussion of tonicity. So those are things that if it benefits you, you may want to review. And that, again, can be found within Chapter 7, because a lot of that information plays a role in how we're able to effectively get rid of waste and also retain things that our body needs. So when it comes to osmoregulation, it's going to be based on creating that balance or homeostasis between the amount of water that the body needs and the amount of solutes. And we know that our cells are working within an aqueous environment. It can be highly detrimental for our bodies to have too little water. If there's too much water, we want to get rid of that too. And the presence of certain solutes within our body, they're playing specific roles. And creating a balance in terms of sodium ions or potassium ions is important, because we have discussed the role that those ions play within events that the cell needs in order to remain viable too. So when it comes to the force that's going to dictate where things are moving, it's going to be all about the concentration gradient, right? And that concentration gradient is going to allow for the movement of solutes across the plasma membrane. So the second bullet point is really just a reiteration of an earlier concept. So we're thinking about concentration gradients. What does my solute look on one side of the membrane versus another? Based on that relationship, what does that tell me about its movement or the need of that solute movement as it pertains to the organism? Here we have some important vocabulary to incorporate into our discussion now. And then whenever you're looking at a particular image as we progress forward, think about how these vocabulary terms can be incorporated there as well. So the major terms are osmosis and osmolarity. And water's movement across the semipermeable membrane is osmosis. So water enters and leaves cells through this mechanism. Then there's osmolarity. And osmolarity is essentially the number of osmotically active moles of solute per liter of solution. And when it comes to osmolarity, we can use terms like hyperosmotic or hypoosmotic or isoosmotic, as you see here. And so if two solutions are isoosmotic, that means they move across the membrane at equal rates in both directions. And this should remind you of the concept of tonicity, which is from Bio 100. And that's the measure of the ability of a solution to change the volume of a cell through osmosis. So examples that we've gone through, if a cell is placed in a hypertonic solution, water will leave the cell and enter the surrounding environment, so it will decrease in size. If the cell is placed in a hypotonic solution, water will enter the cell and it will increase in size. Depending on the cell type, it's possible that the cell could lyse or burst. And if a cell is placed in an isotonic solution, then we would expect an equal amount of movement of water into the cell as well as out, which means that the volume of that cell should not change. So what we're doing right now is, instead of really thinking about it in the way of isotonic, hypertonic, hypotonic, we're now just incorporating phrases like isoosmotic or hyperosmotic or hypoosmotic. But the concept is the same as what you've learned in Bio 100. So we can use this illustration to help support the vocabulary terms that are relevant for this chapter. We have a beaker here. That beaker is utilizing water as its solvent. And then we have solutes that are not specifically described, as far as what they are, but they're incapable of making their way through this selectively permeable membrane that's here. And as a result, we'll have to depend on the movement of the water for an attempt at reaching equilibrium on either side of this beaker. Now, when it comes to this beaker, we can describe the sides based on their osmolarity. So to have a hyperosmotic side, so we're thinking about osmolarity, so the number of active moles of solute per liter of solution, it's hyper, so there's more of it. And we can see that visually illustrated by a ton of circles that are found on the left-hand side. So the hyperosmotic side has a higher solute concentration. And with that higher solute concentration, there is lower free water concentration. And the second part is just hasn't been a way that we've described it in previous iterations of this concept. So that's just something to add in to our discussion now. So we have a higher solute concentration, a lower free water concentration. Now, on the right-hand side, it is hypoosmotic. Again, osmolarity, we're thinking about the number of osmotically active moles of solute per liter of solution. Hypo, underproducing, just in the way that we thought about it for the endocrine system. So there's a lower amount of solutes on the right-hand side and a higher free water concentration. So what we see with our arrow is what we would have seen in our discussion within chapter 7. We're going to have water moving towards the side with the higher solute concentration in an attempt to essentially dilute the left-hand side so that the amount of solutes per volume of water becomes, as best it can, equal to the right-hand side. And now, in terms of a gradient or water flow, what we're seeing here is the movement of water from high water concentration to low concentration of water, right? So down its own concentration gradient, but it's going to head towards a higher solute concentration. So the way that we describe it initially in Bio 100 is based on solute concentration direction. Now we can add in the concept of free water concentration. So just notice here that, again, we're not redefining anything. We're simply adding in additional vocabulary terms so that we can utilize them appropriately for the process of osmoregulation. So when it comes to maintaining a balance between water and solutes, there are going to be challenges that present themselves based on environmental conditions, and organisms will develop ways to overcome or kind of win out over those challenges. Now, when it comes to the mechanism of maintaining that balance, there's two major categories. And one of the major categories is shown here, and that is an osmoconformer. So this is one way of maintaining water balance within the animal. And the osmolarity of body fluids in most marine invertebrates is the same as the seawater. And as a result of that, the extracellular fluids will be isotonic to seawater. So no osmotic gradient will exist, which means in terms of net movement, there's no tendency for water to either leave or enter. Okay, so the rate of movement in and out is such that there's no net movement. And that's a concept that we've discussed, or that is discussed in the first portion of Principles of Biology when it comes to tonicity. Now, when there is no tendency for water to leave or enter, the category is defined as osmoconformer. So an organism that behaves in that manner is an osmoconformer. So they're going to be in osmotic equilibrium with their environment. Now, when it comes to vertebrates, technically only the hagfish is an osmoconformer. Okay, so most marine invertebrates are osmoconformers. And when it comes to marine vertebrates, the hagfish that's shown here on the left-hand side would be an example of like a true definition, like meeting the criteria of how we strictly define an osmoconformer. Now, a shark, on the other hand, would be an example of a cartilaginous fish that is also isotonic to seawater, but it does it in a slightly different way. So it's not just that the osmolarity that we see within the body fluid in the environment are equal to one another. An organism like a shark is doing something specific here. So the way that it's able to maintain that isoosmotic environment is through holding on to high levels of urea. Urea is a type of metabolic waste, and we'll go through the different categories of metabolic waste in a different module. But what the shark is doing is holding on to a ton of urea within its blood. And in doing so, it becomes, for the most part, isotonic to the surrounding seawater. And as a result, there's no net water movement, which prevents water loss. And as a result of this, fish like these don't need to drink seawater in order to maintain an osmotic balance. And their kidneys, their gills don't have to remove large amounts of ions from their bodies because they're not taking in the surrounding seawater. And when we think about how they're able to maintain this isoosmotic environment, we should factor in or consider the fact that they have a ton of metabolic waste in their body. And we know when it comes to excretion that we want to get rid of the waste that we generate. And so what we're able to kind of learn from this process of maintaining this isoosmotic environment is that organisms like the shark have enzymes in tissues that are really tolerant of high levels of this nitrogenous waste. And that's what allows for them to utilize this mechanism to kind of operate under the category of an osmoconformer. Here we have the category of an osmoregulator. And osmoregulators control their osmolarity internally, and they'll maintain a pretty constant blood osmolarity, even if they're in different surrounding environments, like the salmon here, which would migrate between rivers that have a lower solute concentration versus oceans, which would have a much higher one. And when it comes to osmoregulators, a lot of marine vertebrates and some marine invertebrates are osmoregulators. And we'll go through the idea of osmoregulation in kind of the marine environment versus the freshwater environment in our upcoming slides. In this illustration, we'll go through how osmoregulation is achieved in a marine fish. Now, if we think about our vocabulary terms, for marine fish, they will be hypotonic relative to their environment. So their body tissues, extracellular fluid will be hypoosmotic compared to the seawater. So that would mean if we are thinking about what water would want to do in that situation, if the marine fish is hypoosmotic, then it has fewer solutes within its body, which means that outside there'll be far more solutes. So water found within the fish will want to exit towards the higher solute concentration, and that will take place through osmosis. Now, this would not be beneficial because if we think about a ton of water moving out of the marine fish, then it's possible that it can result in desiccation or extreme dehydration, excessive water loss, which is not going to allow the fish to be viable. So how does the marine fish adapt to its surrounding environment? So the marine fish will respond by taking in large amounts of seawater and eliminating the excess ions through their kidneys or through their gills. So if we look at the illustration here, we have the fish taking in through drinking water. Okay, so by taking in the water in the surrounding environment, there has to be the ingestion of salt ions too, because you can't partition out one versus the other. And in this illustration, the lighter arrow is water, the dark arrow is salt. So the fish is taking in and ingesting a ton of water and a ton of salt. But we want to maintain a balance of both water and solids. So there needs to be some loss here. So after ingesting a ton of water and salt, there's going to be excretion of salt ions through the gills and osmotic water loss through the gills and other parts of the body surface. And when it comes to excretion, right, the elimination of waste, that's another opportunity for salt ions to leave the body. But there will only be small amounts of water that leave. So scanty urine means small amount. So the fish is able to get rid of that salt and hold on to a pretty decent amount of water. So it doesn't become desiccated. It's taking in water and then getting rid of some of the salt so that it doesn't have too much in order to maintain that balance. And so we can see within the urine of a fish like this that there will be small amounts. And when we think scanty urine in this scenario, we're thinking about preservation of the water that it ingested initially. So this is a summary slide that is from the Campbell book based on the illustration that was previously shown. So we have the marine bony fish, and the environment is far more concentrated than the body fluids. So the body fluids are hypoosmotic to the surrounding environment. And as a result, fish would lose water. Okay, so there would be a loss of water as the water that's within the fish wants to leave. But there can also be a gain of salt, right? Because if it's hypoosmotic, that means that there are less solutes in the surrounding environment. And so the salt that's in the surrounding environment would go down its concentration gradient and enter the fish as well. So as far as inflow, outflow goes, the fish again is going to drink water, and as a result, it's going to be taking in salt too. So it drinks water, so salt's going in, okay? And salt's going to go out eventually through active transport by the gills. And if we're saying that active transport needs to be utilized, we're saying that the difference in terms of solute concentration is going to be lower within the fish, higher in the surrounding environment, because we're going against the concentration gradient when we utilize active transport. And there will be some water that goes out. And it looks to me like they're still using the same colors to represent salt and water here. Now, in looking at the urine of the fish, it's small, so that was the scanty description in the earlier slide. And the urine that's produced is slightly less concentrated than the body fluids. So I want you to think about what we're saying in terms of adaptations for osmoregulation. And then as we go through the actual organs that are involved in this process, then think about how that relates back to this initial discussion. Here we have an example of osmoregulation in a freshwater fish. So a freshwater fish is going to be in an environment with a really much lower amount of solutes compared to marine. So a freshwater fish has a significantly higher solute concentration in its body fluids than that of the surrounding water. This makes the fish hypertonic to its environment. If the fish is hypertonic to its environment, meaning that it has a higher amount of solutes within it, then water is going to want to enter the body of this fish. So the goal is to take in water for this fish, but also release the water as dilute urine, which helps get rid of the excess. Okay? So freshwater fish will have a tendency to lose some of the ions that it has, but it can actively transport ions back into its body. So in this example, we're seeing that there's a gain of water because water wants to enter the fish, okay? And some ions in food. So notice that the fish is not actively drinking in the same way that the marine fish was, because it doesn't really want this water. It doesn't need it. Now, in thinking about kind of the solute movement, we'll see an uptake of salt ions by gills. We'll see osmotic water gain through gills too. Okay, so there's some acquisition of water and solutes through food, not through actively drinking. And then there's some gain of water and salt through other mechanisms. So osmotic water gain, as well as uptake of salt ions by the gills. Okay, so we have water and ions coming in. And in response, the fish is going to create a really large volume of urine, and that urine will be dilute. So meaning that it's not going to have a ton of solute in there. Okay, so again, the desire for the freshwater fish is to hold on to its solutes. So it doesn't want to lose them. It's still going to lose a little bit, okay, but it does have a means to actively take ions back into its body. So this allows for the freshwater fish to maintain a balance. So large amount of urine, but that urine will be dilute with respect to solutes. And then this scenario, we're just thinking about salt ions.